Hello again, everybody. Welcome to Virtual Lab 11. Today we'll be talking about sequencing technologies, and then we'll start some bioinformatics things that will springboard us into next week, where we will do some more in-depth analyses. Everybody say hi to Pancho. I started talking and he woke up from his nap to come say hi. Okay. So we'll talk about Sanger sequencing, Illumina sequencing, and then databases and different bioinformatic analyses that you can do just on the introductory level. And then again, like I said, we'll go a little more in depth next week. Okay. So to start out, let's look at some nucleic acid sequences that we have. So these are different types of nucleotides. And so if you look over here on the right, I have laid out a ribonucleic acid, a deoxyribonucleic acid, and something called a dideoxyribonucleic acid. So the difference between these is going to be in the, let's see, 1, 2, 3, 4, in the third and fourth position on the sugar, you'll see that there are some hydroxyl groups sticking out. So the difference between DNA and RNA is the presence of two of these groups or of just one of them. So that's why it's called deoxyribonucleic acid, because it's a ribose, but it's deoxygenated on the second carbon. And so you'll notice this one has two oxygens, or two hydroxy groups, and this one only has one. So with that in mind, look at this particular string of nucleic acids and tell me what kind of sugar base is it? Is it DNA or RNA? And obviously you can see that because each of them has two hydroxyl groups, it's RNA. Now I want to look at dideoxy. So di meaning two deoxyribonucleic acids. So that means both of these groups are just hydrogens. They don't have any oxygen coming off of them. And this will become important in just a moment. So let's look at synthesis of a DNA strand. Typically what will happen, and so again, this particular strand, is this DNA or RNA? And the answer is DNA, because it's deoxyribose, because it's deoxygenated on the second carbon on all of them. So they only have one oxygen. So typically when you're synthesizing a DNA strand, what will happen is that the oxygen, the OH group on the sugar, will attack the phosphate group of the nucleic acid that is being added onto or synthesized onto the strain through a condensation reaction. Don't worry so much about the organic chemistry of it. What I want to point out, though, is that this oxygen is required for synthesis of a strand onto it. And so what will happen is that it will attack it, and then it will actually kick off one of the oxygens in the phosphate group. So this isn't totally accurate, and it's also not a double bond. You can see each of these has a double bond, but for our purposes, it's close enough. Okay. So now I'm going to synthesize another one. Same thing, the oxygen group is going to attack the phosphate, and that will allow synthesis, an addition of the nucleic acid onto the strand. What do you notice about this particular nucleic acid, though? It's a dideoxy nucleic acid. And it doesn't matter which base it is. There's pyrimidines and purines. Here I didn't label them because it doesn't really matter for this right now. So this is dideoxy, meaning that it is lacking two hydroxyl groups. So what does this mean when I try and add on another nucleic acid? What will happen? What kind of reaction will occur? Or better, what kind of reaction won't occur, and why? The answer is that without the hydroxyl groups, that there is no oxygen capable of replacing or attacking this phosphate group, and so nothing will occur. So it will actually not be added onto the strand. When this occurs, it's called chain termination. Chain termination because synthesis stops with the dideoxyribonucleic acid. Okay, so let's talk about how we can use the chain termination technique to sequence. So I have up here a 12 nucleic acid strand. It's just random letters. The ends on either side are irrelevant for our purposes right now. I just want to show you that we are only interested in sequencing this part of, or we're only interested in obtaining the nucleic acid identities of this particular sequence, and we're not going to care about the preceding or succeeding ends that are just random nucleotides. Okay, so we have our sequence here. It's ATG, CGC, ATC, TAA. And what we're going to look at is how to sequence it. So think back to PCR and some of the required reagents to synthesize a new DNA strand. So you have to have your polymerase. That is what synthesizes the new nucleic acid onto the old ones. You have to have your buffer, a salt buffer, make sure that the polymerase is happy. You have to have a template strand. In our case, what we're doing right now, the template strand is this. So we're going to have a bunch of copies of this particular segment of DNA. The next thing you'll need is primers. Again, in this particular example, the primers don't matter. It's going to match this sequence and this sequence, so that here, I'm going to clean this up. So it'll match this sequence and it'll match this sequence, so that when I extend using the polymerase, it can go in both directions. So yeah. And the last thing is we need free nucleotides to get added on to the strand to extend it. Okay, so I have 4 tubes here, and each of them I'm going to add a special ingredient. In this case, it's going to be the dideoxynucleotides. So DD stands for dideoxy. In this case, it'll be adenine, cytosine, guanine, and thymine. Oh, you can't see that one. Okay. So that means most of the free nucleotides will be the deoxy NTPs, so the DNA equivalent of the DD, ATP, DD, CTP, etc. There's a low concentration of each of these, but at some point they're going to get incorporated by the polymerase into the strand. Remember what happens when I incorporate a dideoxynucleotide? It terminates the strand synthesis. It will prevent addition of additional nucleotides. Okay, so starting again, looking at this particular sequence, if I'm going to try to synthesize these 12 nucleic acids and I incorporate a DD-ATP, so I get these first 5, and then at this spot the DD-ATP is incorporated, what will the strand be? What will the sequence be? It'll just be A. And again, there will be N's before it, but I chose not to include those just to keep it simplified. So it will stop at the A. Okay, so what happens then if I incorporate a DNTP at the first site, and then I continue on? Where will the next synthesis site terminate? Like, where will chain termination occur if I'm looking at only these 12 nucleotides? Remember, only these 12. So remember, in this particular tube, I have DNTPs as well as DD-ATPs only, so that means that chain termination has to occur at which nucleotide? It has to occur at the A, because this is where synthesis, that's supposed to be a star. This is where, if I incorporate an A and it's a dideoxy, then it will terminate the strand at that location. So if this is the first possible place of termination, then the second possible place of termination in this particular one is at this A right here. So it can be that possible sequence. If I incorporate a DNTP, so a deoxy ATP at this particular location, but I incorporate it at the next possible location, what will the sequence be? So if I follow along, it'll be ATG, CGC, A, TCTA, and then finally, the last possible place that I can cause chain termination with A's is at the full sequence length. So after all 12 nucleotides have been incorporated. I'm also including this sequence with this N right here to indicate that chain termination occurred after the 12 nucleotides of interest have all been synthesized. Okay, so just to repeat what I just went over. So when I incorporate a dideoxy nucleotide, in this case DDATP, so anytime there's an A in the sequence, if I incorporate the dideoxy nucleotide, chain termination occurs and I cannot synthesize beyond that site. So in this particular sequence, I can chain terminate at the first A, the second A, the third A, or the fourth A. Or I can not incorporate the dideoxy ATP at any of these sites and actually terminate somewhere over here further on beyond the sequence of interest. So that is where these 4 possible sequences come from, is that I can be synthesizing a strand and terminate at one of the 4 locations. Okay, so now let's move on to DDCTP. So if I have dideoxycytosines, everything else is normal and there's a low concentration of these, so this only gets incorporated sometimes into the strand. Where are the possible sites that I can terminate? So the first one, so again, looking at this particular sequence, the first place that I encounter a C is right here, which means that I can get ATGC. If I incorporate a deoxy NTP at this site and continue on until the next possible site is going to be right here, which means I can get this strand. And then further, this next one is this one. And then again, I included this one to indicate that if all 3 sites, so there's 3 possible chain termination sites with DCTPs, if I incorporate DNTPs, sorry, I said that incorrectly. If I incorporate DCTPs at each of these 3 locations, then strand synthesis will continue and I will get the full 12 nucleotides. Plus, again, the irrelevant part on this side that I don't care about for right now. Okay, if I do the same thing with DDGTP, where are the possible sites that this could cause strand termination? Well, it's everywhere there's a G, so it could be here, and that's actually it for that particular one. And then again, this one, if both sites incorporate just regular deoxyguanosine triphosphate, so DGTPs, then the entire strand will be synthesized, and that's, again, this is what this N represents. The last thing is going to be the DDTTPs. So everywhere I have a T, those represent possible sites that chain termination could occur, and so there's 3 possible sites there. Okay, so again, just to go over what I was just talking about. So if I want to synthesize a strand, I can incorporate what are known as dideoxynucleotide triphosphates. And when I do that, strand synthesis terminates, also known as chain termination. And so anywhere that I incorporate a dideoxynucleotide, chain termination occurs. Okay, let's talk about how I can use this for sequencing. Oops. So let's arrange these in size order. Okay, so of the 12 nucleotide sequence that I'm interested in, and again, there are ends before and after the sequence that I don't care about, and we're not going to worry about these right now. So of the 12 nucleotides, here is starting from the first nucleotide to the 12th nucleotide are all of the possible sequences that I can get. Okay, so I just moved that over here to the left, and we're looking again at all of the possible configurations. If I incorporate DATPs, remember all other nucleotides are just single deoxynucleotide triphosphates, which means that termination can only occur in the tube where I'm using the dideoxynucleotide. So in this one, it's ATP, DD-ATPs. This one is DD-CTP. This one is DD-GTP, and this one is DD-TTP. And this is useful for me, as we'll see moving forward. So let's take then, in this tube, these are all the possible sequences that I could get, and et cetera, in these other 3 tubes. So let's run these on a hypothetical gel electrophoresis. Okay, so if you count each of these bands, there are 12 of them. Looking at these bands, and remember, the shortest sequence length was 1 nucleotide, and the longest sequence length was 12 nucleotides. Which of these bands represents the single nucleotide, and which of the bands represents the 12 nucleotide sequence? And why? So this will be the 1 nucleotide band, and this will be the 12 nucleotide band. And this was something that I covered in last week, because gel electrophoresis separates out sequences by, or like segments by size. And so the shorter it is, the further it will run down the gel, because it can get through it easier. It's a physical process. If it's longer, like the 12 nucleotide sequence up here, it will not travel as far, which means that this one, if we're comparing relative lengths, this one's 11, this one's 10, this one's 9, 8, etc., down through the gel. And so I've labeled them here. I didn't include a ladder in this particular gel, but that is something that should always be done. So again, they're arranged by size order through the gel. And one thing that is worth mentioning is that each of these bands, because anytime we do anything with DNA, you have to do it in billions of copies. If I use one single DNA strand and I run it on a gel, I won't be able to see it. It will just be too faint. So even if I had a 1 right here, I wouldn't be able to see it. And so each of these actually has millions of copies of single nucleotides that we're able to run down the length of the gel. And similarly, this particular one has millions of copies of the 12 nucleotide sequence that I can see together as a band on the gel. Okay, so think back to the particular sequences that we were looking at. I arranged it by size order, and obviously you didn't memorize the sequence, that's fine. But let's look at what each band represents. So remember, the shortest possible one was this single nucleotide A, and the longest possible one was the whole 12 nucleotide sequence length. Because each band represents a possible site of chain termination, I know that it can be used to represent a nucleotide at a particular site. So I know that because this band down here was 1 nucleotide and it shows up in the A column, I know that this must be an A. If I find the 2 nucleotide length one, which is this one over here, and again, here's the numbers for reference, which column did it terminate in? It terminated in the DD-TTP column, which means it has to be what? At the second site. It has to be a T. And this agrees with our sequence. Same thing with the third one. The third one terminates in the GTP column, in the DD-GTP column. So that means the third nucleotide must be a G. Let's see, 12, 11, 10, 9. So if we look at the 9th band up, so again, looking at the numbers, here's number 9 right here. This terminates in the CTP column, which means that the 9th position must be a C. And again, this agrees with our sequence. And so this is Sanger sequencing. This is how it's done, is you can get a template strand of DNA, and then you can synthesize copy strands of it using these dideoxynucleotides that terminate at specific sites. And if I keep the samples separate, I have all of the bands in this lane terminated with an A, and all of the bands in this lane terminated with a C. This lane was a G, and this lane was a T. And so the next thing you can do is you compare them by band length, which again is where these numbers come in handy to say, okay, this is the shortest possible one where termination occurred, which means it's one nucleotide at two nucleotides is over here, 3, 4, 5, 6, 7. And then you can determine which nucleotide caused the chain termination by which column it's in. So again, that's where the A, C, G, and T comes in handy. Okay. Oh, and so if I want to get the sequence, so this is what the actual sequence was. If I eliminate all of those extra letters, I can say, okay, site 1 was an A because it terminated in the A column, site 2 was a T because it terminated in the C column, and this is actually provides from the bottom up the identity of your sequence. So ATG, CGC, ATC, TAA is the same sequence that was provided in the previous slide. Okay. So here is a second method that is used in Sanger sequencing. Rather than keeping the columns separate, so the A's, C's, T's, and G's in different columns, I can actually put fluorescent molecules onto the dideoxynucleotides. And so in this case, G's will show up green, A's will show up yellow, C's will show up orange, and T's will show up red. So now if I put these all in the same well and run them again on a gel electrophoresis, this is what it will look like. And so now I can tell by color the same information that I was able to tell previously. And so starting from the bottom, if it shows up yellow, I know that A was A at the shortest location, so this is going to be one nucleotide in length. It's yellow, which means it's an A. At the second nucleotide position, it's red, which means it's a T. And then green is G, orange is C. And so, again, if I were to do these again, it would tell me the identity of the 12 nucleotide sequence based on the color of the band, starting from the bottom up. So it'll be A, T, G, C, G, C, A, T, C, T, A, up at the top. And so it's the same idea, it's just these are color-coded rather than having to go between multiple lanes to determine like how the sequence is arranged. And so that zigzag pattern that I just did was getting from basically moving from size up from the shortest up to the tallest one. But in this one, it's a little bit cleaner again because it's all in one lane and I just look at the colors to determine the sequence length. So this is Sanger sequencing. This is how it's done. It was determined, I think, is either late 70s or early 80s that this method was or that it was created by Sanger. That's the name of the person who did it. And this was how sequencing technologies began. And so I have a couple examples up here. So this is a picture of an actual Sanger sequencing gel of what it actually looks like. So you can see that, remember, the sequence identity, you start from the bottom and move your way up because the shortest ones are at the bottom. So that means chain termination occurred earlier on than the ones that are up at the top. And so again, if you follow it along, you can see that the sequence, you sort of have to follow like a zigzag pattern to determine the length. Oops, see, I missed one there. It's a little difficult. And so again, my cursor is just following the size of the bands. And so using this up here, so this first lane, this is a T, so then A, then T, then A, then T, and then G, G, C, G, T, T, etc., to determine the actual sequence length. And so I have a couple of examples here. You should pause the video to practice these. This one right here, and then also this one right here. So again, these are the two methods of Sanger sequencing. This is chain termination, and then this is the fluorescent chain termination. So pause the video here and determine the identity of the sequences that I have presented on these gels. Okay, so the sequence in this one, so what nucleotide is it going to start with? It's going to start with, remember, the shortest band where termination occurred first is going to be right here. So it's going to start with a T. And then what is the last nucleotide in this sequence? That's going to be where the band is the longest, so also with the T. And so if I were to figure this out, so T, and then the next one is A, and then G, A, G, A, C, C. So this ends up being the sequence right here, based off of the bands. Okay, what about this particular one? This is using the same colors as are shown right here. That's supposed to be an arrow. And so green, again, that's G. Blue, no, I said that backwards. Green is A, blue is G, pink is C, and then A, C, T, G, T, A, G, G. And so that is the sequence for this particular column. So some of the benefits of Sanger sequencing, again, this was the first sequencing method that was used, going from not being able to determine a nucleotide sequence to now we can sequence things and start like actual genetic analyses based on them. I mean, there were ways to do it before that are actually quite innovative, but this really revolutionized our ability to, it basically was the birth of bioinformatics. And so it's relatively cheap. We can do probably 1,500 nucleotides at a time. And so imagine doing a Sanger sequencing with 1,500 nucleotides. It's just a super long gel with just, 1,500 bands at all different locations. And it looks like this particular gel, just very big. And so we can do a Sanger sequencing analysis for probably, I think, $20 per sample. So that's pretty cheap. And it's pretty fast too. You can usually get it within a day or two. The other side of that, though, is it's pretty inaccurate on the edges of the sequence. So the 5' and the 3' ends are going to have pretty high inaccuracy rates. And that's just because gels get a little fuzzy on the tops and the bottoms of them. And the other thing is that we're limited to how much we can sequence from them. Usually the max you can do is, I don't know an exact number, but I would say probably 5,000 is probably the highest you could go comfortably. The best thing to do, though, would be to chop that up into smaller bits of 1,000 maybe and do 5 1,000 nucleotide segments. So that is Sanger sequencing. It's very low tech, and we still use it, but it is not the best way to sequence. Well, not always. Depends on what you're doing. The next thing we'll look at is Illumina sequencing. This is also known as next generation sequencing because it is a little bit more high tech. There are many variations on it. There's dozens, if not hundreds, of sequencing technologies now that are all some variation of this particular sequencing method. Illumina is a specific company, and there are other companies that have their own methods, but Illumina is by far the most used sequencing technology. The benefits are it's extremely accurate. You can do very large sequences. So like the Human Genome Project that was done, any type of human sequencing now, so 6 billion nucleotides are done using Illumina. So you can get very, very large sequences, and you can do massively parallel sequencing. And this just means that you can do many sequences at once. And I'll use some numbers to clarify that in a minute. But suffice it to say that you can do up to several hundreds of different sequences at one time. and it's also very quick. You can usually get these results usually within 48 hours. The pitfalls are obviously it's going to be expensive to do any sort of large data analysis like this. And because it's massively parallel, you're going to get extremely large data sets, and you have to have a good computer and good software to process it. Otherwise, the sequencing itself is useless. And I'll talk about how to process it in just a second. Okay, so these aren't, I have, I think, 9 steps in this process. These are just like general steps. It's not like a super hardcore protocol or anything. So the first thing you'll need to do is you need to get millions of copies of your sequence of interest. And so if I'm going to sequence, let's say these are 3 bacterial genomes, I'm going to need to get millions of copies of them in order for this to work. And so, again, millions of copies so that I can sequence them. So if you are growing, or if you're wanting to sequence a bacteria, think of how you can get millions of copies of a bacteria. What would you do in the lab to make sure you get a lot of copies of a particular genome? So the way that we typically do it is that, remember, in a liquid culture, you can get up to like 1 million cells or more per milliliter. So if I want a lot of copies of the genome and every cell has 1 copy, then I can grow, let's say, 10 milliliters of my sample, and then centrifuge them to pellet the cells, and now that pellet has billions, if not trillions, of copies of the genome. Okay. Yes, lots. Okay. The next thing that has to happen is you fragment the DNA. So you cut it up into bits of the same size. And there are different ways to do this. You can use what's called sonication, which uses sound waves to chop up the DNA. You can use like a nebulizer, which forces it through a small hole, which will shred the DNA. You can do it enzymatically. But in any case, what you have to do is you have to get segments of the DNA chopped up so that they can be processed. And remember, this is a random process. And so if I have, so like just looking at this red one, at the red genome, if I have 1 million copies of it and I'm shredding it into 250 like 250 to 500 nucleotides. Sorry, this is sort of a slow process. Ooh, okay. So if I'm shredding it into 250 to 500 nucleotides, I'm going to have a lot of copies that represent the full genome cut at different sites. And that is good. That's what we want. We need a lot of random 250 to 500 nucleotide segments of the genome. And that will make it easier later. So you fragment the DNA, and then you do what's called tagging with an adapter sequence or a barcode sequence. So let's look at that for a second. So these are barcode sequences that get added on to the end of every single fragment. So millions, billions, and or trillions of these fragments will get these adapter sequences on them. If I keep my samples separate, then I can add them. I can add different adapters onto different samples. So if this is sample A, it will get the orange adapter. Sample B will get the yellow adapter. And then sample C will get the purple adapter. So let's look at what the adapter is actually made of. So on average, it's about a 35 base pair or 35 nucleotide sequence that I know the identity of the sequence. I know what the sequence is. I know the order of the nucleotides. The outermost 10 nucleotides, and these numbers do vary depending on the technology and the kit that you're using, but the outermost 10 will serve as a primer binding site. So when I do PCR later on, I can add primers that will then extend the sequence. The colored region is 25 nucleotides identifier sequence. So this is what's the barcode region. So I can make those 25 nucleotides any nucleotide that I want, and it will be specific to the particular sample that I'm looking at. And so again, when I'm looking at these three, the black sequence is the primer sequence found on all of the barcodes or all of the adapters, and the colored region is what actually provides the quote unquote barcode for each sequence. So all of sample A is going to, again, it will get the orange sequences, which will have a unique 25 nucleotide sequence, as compared to the yellow one, which will have a completely different one, which will have a completely different one from even the purple one. And this is useful later because everything gets mixed together, and that can get a little confusing. So once they get mixed together, I PCR amplify them. And so again, using the black sequence, the 10 nucleotides I know, I will create a lot of repeat segments of each of the samples, both the green, blue, and red segments. And keep in mind that this is occurring at many sites of the genome. So it's not just this particular one site. This is only 250 to 500 nucleotides. If I have the whole genome and I chopped it up randomly, well, that's just this particular copy. I also have 1 million copies of the same genome that is going to be cut in different regions. And each of those is going to get an adapter sequence added onto the side of it, onto either end, the 5' and the 3'. These are double-stranded sequences. Okay, so I PCR amplify them, so I have lots of copies of them, and then I flow them through what's called a lane. So in Illumina sequencing, these are lanes that are used to actually do the sequencing, to carry out the reaction. And so each of these, you can see them separated by like a black strip. Each of these is their own individual lane. And so this particular one has 8 lanes on it. This whole thing is the size of a microscope slide. Okay, so what the lane will do is that it will have, so this is a bird's eye view of a lane. So you'll see that I have these black squares. These represent the primer sequence. So it's the same sequence as are found on the adapter sequences. So that when I use a pipette and I put the samples through a lane and I run them down the lane like this. As that happens, the primer sequence on the end of it will actually bind to the primer sequences attached to the lane. These are covalently bonded to the glass slide. So these particular ones do not go anywhere. They are stuck to the slide. And so as a complementary sequence flows over it, it will bind to it just like it would a primer in a test tube. So we got that. So that's sort of what it looks like. And so we'll change the view and do what's called bridge amplification. So this is the same thing. This is still the lane. I flowed them through the lane this way, and they anneal to the primers. Now they are bonded to these primers that are stuck on the glass slide. Bridge amplification is a process used to amplify each particular segment of DNA so that it creates millions of copies at that particular site. So I just want to, again, instill upon you the large number of sequences that are currently being analyzed in this process. So each of these represents a 250 nucleotide. Each of these represents a 250 nucleotide segment of a full genome. So again, my genome is chopped up like this into 250 nucleotide segments. Well, remember, I had billions of copies of my DNA sequence that are all chopped up now in different locations. And so eventually the entirety of the genome will be represented by these 250 nucleotide segments. And so that's why now that I have millions of copies of them, this will help me in my next step. Okay, the next step is fluorescence sequencing. So I've zoomed in on just a single strand of the genome of interest, or the segment of the genome of interest. And now to actually do the sequencing, what they will do is they'll get a DNA polymerase that will amplify this particular segment of DNA, and as it does so, it will incorporate fluorescent nucleotides. So if you remember back to Sanger sequencing, the A's were a particular color, the G's were a particular color, C's and T's each had their own color. It's the same idea here, except that as it gets incorporated, it flashes. So I have a camera here. It's a very high-tech camera that is capable of seeing these flashes on a microscopic level. But if you remember, all of these clusters are the same sequence. So if I do 1,000,000 at the same time of just like, let's just look at this one. If I incorporate A's at 1,000,000 sites all at the same time, it's all going to flash red at the same time. And so the camera will be able to see that. It is a very high-tech camera, and I don't want to spend too much more time on this. I've included some videos about Illumina sequencing from the actual company that will explain this a little more in detail that I want you to watch. And so the DNA polymerase, it anneals to the sequence, and as it goes through, these flashes occur that are picked up by this camera. And then it is able to say, okay, it flashed red, which is an A, it flashed yellow, which is a T, it flashed green, which is a G, and it flashed blue, which is a C. And so then it will output this sequence that I can then use to analyze. So remember, on the lane, this is occurring millions of times, and so every time a nucleotide is incorporated at each of those clusters, it will flash a particular color. And so this camera is able to keep track of every single site on the lane. And so I'm going to go back for just a second. Okay, so this particular lane, so there's 8 lanes on here. In each of these 8 lanes, you can put up to 99 unique bacterial samples. So that means you can get 8 times 99 completely unique genomes. And once they're all getting sequenced at the same time, this camera is able to pick all of those flashes up and basically say, okay, at this location it's this, at this location it's this sequence, at this location it's this sequence. And so it's very high tech. Okay, so looking at this particular sequence that was output, now I can use that for sequence assembly. So this should look familiar. So again, here's my adapter sequence on either side of my nucleotide, unknown nucleotide. So I'm putting, oh, well, it's already labeled unknown sequence. I can say, okay, it flashed in this particular order, which means I now know this particular sequence. If I look at this sequence, hey, they overlapped right here, which means these probably align. And now I know this sequence right here aligns. And if I say, hey, this sequence aligns with this sequence up here, now I can say this is my unknown sequence, but hey, it aligns with this sequence up here. And so using that kind of overlapping method, using all of these sequences, you can gain what's called a consensus sequence. That's an E. All right. Consensus. A consensus sequence, which is basically the identity of the original genome. By sequencing all of these fragments individually, I can now get a really sophisticated computer program that is able to say, hey, these all align at this particular site. Like at this particular site, these are all A's. At these particular site, these are all G's, which means I know that they align and they overlap. And even though I reach the end of this particular sequence, this one provides the rest of the genome, or the rest of the sequence at that site of the genome. And so like all of these align right here. And so I can say this is the consensus sequence at this location, because I had all of these fragments line up right here that gave me the same nucleotide strand. Okay. So that was Illumina sequencing. Again, I have included on the worksheet and then on the announcement that I will post, there will be links to a couple of YouTube videos about both Sanger and Illumina sequencing that I want you to watch, again, to just hear it from somebody else. But this was a good introduction. Okay, the last thing we'll talk about today is bioinformatics, part 1, and again, part 2 will be next week, where we will do some sequence analyses more in depth. So bioinformatics is the science of collecting and analyzing complex biological data such as genetic codes. It's also known as in silico analysis. So in silico just means in the computer, basically. Okay, so some examples of that. So again, sequence analysis, so post-sequencing, analyzing the sequences and comparing them to one another and to known sequences. RNA sequencing, um, phylogenetic analyses, molecular interactions and systems biology, so how basically using supercomputers to determine active sites of proteins and like this is good for like how medicines work, how it can inactivate a particular enzyme or how it could kill a cell, etc. Structural analysis, same thing, back to like protein structures, that type of a thing. Database manipulation, we'll talk about a database called NCBI, so National Center for Biotechnological Information, and how we can use that. Okay, so some terminology to get started. So I will take you through several terms, and then we'll go to the database website, and then I'll show you how to kind of navigate through it a little bit, and then the lab report will have you do that on your own. Do some of your own investigating. Okay, so when I say alignment, this is a base by base comparison of two sequences. So it can be either amino acids or nucleotides. And so this is just me aligning them to see how similar they are. So if I have ATGC, I can align it with a different sequence that may be ATGG and say, hey, they're different on the last nucleotide. Same thing with amino acids. Percent identity is the number of shared bases or amino acids in an alignment. So basically, how similar are they? Are they 99% similar? Are they 50% similar? Are they 2% similar? Based on the alignment. So query and subject are two terms that you will see a lot in databases. So if I have a sequence and I want to compare it to other known sequences, the sequence that I am asking about is the query. So it's the sequence being tested, and anything that it aligns to then becomes the subject. So the subject is the thing that the query aligns to. Query cover is the percentage of the unknown sequence or the sequence being tested, how much of that sequence is covered by the subject. So if I, like, let's say I have this particular sequence, so this will be my query, and I want to align it to this sequence as my subject, well, you'll see that the subject sequence is actually shorter than the query cover, so it may only be 80% query cover. Okay, so that's that. The last thing is the e-value. So this also stands for expect value, and it is a statistical analysis, basically telling you how accurate or basically the likelihood that your alignment is due to random chance. So how many times you expect to see a similar alignment due to random chance. And so if you are familiar with the p-value from statistics, it's very similar, just used in the context of databases, specifically for bioinformatics. And so it helps you determine if two sequences are related biologically. And so keep this in mind. So the e-value, we'll come back to that as I go into the database, but I just wanted to introduce what it is to you. Another way to think about it is asking what are the chances that it's a false positive. And so it will give a number, and it will basically be, you can think of it as like a fraction or a percentage likelihood that it is due to random chance that I see this alignment. Okay, some additional terminology. I'll go through these pretty quick. So CDS, you'll see, is the coding sequence. It's the nucleotide numbers of a particular gene. ORF is open reading frame. In a sequence, you will have a variety of ORFs, and it just corresponds to the gene number. So ORF14 is the same thing as gene 14. UTR is untranslated region. PP is polyprotein or polypeptide. A mature peptide is abbreviated as MATPEP or MATpeptide. We'll see this in just a second. If I have a polyprotein, the mature peptide is so this is a polyprotein. If I cleave it at specific sites, the mature peptide is what results from it. So these individual segments will be the mature peptides that then have their own functions. A locus tag is a completely arbitrary name for a specific gene in a sequence. And again, this will all make more sense once we're actually looking at it. The product is the function of a gene or the name of a gene. A protein ID is a unique identifier for a particular protein product. And frequently it will provide a link in the database to another database where you can look at that particular protein of interest. NSP is a non-structural protein, and a GP is a gene product, also known as a protein product, otherwise known as the protein, the translated gene is what the gene product is. Okay, so now we're going to go to NCBI and actually look at some things. So this is the website, it's in the PowerPoint. You can also just Google it, it'll show up, but it's the National Center for Biotechnological Information. Okay, so first thing we're going to do is we're going to look at the SARS-2 coronavirus genome. I'm actually going to research that for you. So SARS-2 complete genome. If I type in any organism and then followed by complete genome, usually the first thing to show up will be what I'm looking for. If you have never heard of NCBI, this is a really, really great place to do research, like analytical research on papers that have been done. And so this will provide access to a lot of papers in every type of biology. And so it's just a really great place to look for new information. So if I ever have to learn something, the first place I go to is NCBI to type in, like, let me think of something. Like if I want to look up how an antidepressant works, I would do like SSRI mechanism of action or something like that, and then it will provide me, I will usually go to PubMed, and that will provide 32,603 papers about SSRI mechanisms of action. Okay, anyways, so right now I want to look up the severe acute respiratory syndrome coronavirus 2 complete genome. If I search that, it will usually come up first thing here. So when we talked about it in class, I said that there were 8 proteins. There's actually 12. And so this is an overview of what the SARS coronavirus 2 genome looks like. And so here you can see the polyprotein. There's actually 2 polyproteins that are used by this particular virus. And then here's some structural and non-structural proteins. So I'm going to go ahead and click on it, and it will take me to a new page where I will have all of this text information. Most of this, for our purposes, doesn't matter. It's just about the authors, like where they obtained the sample, etc., how they sequenced it. And so you'll actually see, so the sequencing technology that they used is actually Illumina. And so that's how they got this particular sequence obtained. All right, so remember, as I'm going through this, you're going to see a lot of the things that I just mentioned at the end of the PowerPoint. So the 5' UTR, so the first 265 nucleotides are part of the 5' untranslated region of this virus. Then the first gene begins at 266 and is actually the first 20,000 nucleotides. And so the translation of those first 20,000 nucleotides are this giant protein right here. And so the virus will actually translate the RNA to this huge polypeptide, and then it will cleave it at specific sites to make the mature peptides. So this is what I was talking about. So ORF1AB, so that's the name of this huge gene up here. And then it will cleave it at different sites to make all these different mature peptides that each have their own specific function. So that's that one. There's actually remember that second polypeptide. It's not quite as large, but it's still quite large, and it has its own mature peptides that it uses. Sometimes it will include things like the product, like I was mentioning, is the name or the function of the protein. So this particular mature peptide, here's the sequence that it's the nucleotides numbers that it's found between. Here is the name, the gene that it is named from or found in the sequence. The locus tag, again, this is arbitrary, and this completely depends on the organism that you're looking at. And so it's part of gene product 1, which is the same thing as ORF1. So open reading frame or gene product 1 are the same thing. It is a 3C-like proteinase. So that just means that a proteinase is an enzyme that cleaves protein. So this particular protein, this particular mature peptide, may play a role in cleaving the other mature peptides later on. Oh yeah, look, mediates cleavages downstream of non-structural protein 4. Anyways, and then, so yeah, sometimes the notes will include additional information. So what I'm showing you then is just basically the actual sequence of this particular virus. And so further on, there's those shorter segments of the protein. So if I go back, you can see there's some short segments here. So going back into the genome, scroll back down to the bottom, and then I get all these shorter polypeptide sequences that it uses. At the very bottom, it has the nucleotide sequence of the full virus. And so there's quite a bit of it. So there's, again, almost 30,000. But these are the actual nucleotides, and the order that they're in that creates the virus that is now currently spreading around the globe. Okay, and then there's also a 3' untranslated region found at the very, very end of this particular virus. Okay, so what can I do with this information? So let's go down and find one of the shorter proteins. This is a good one. So it's like a medium length one. So it's pretty long. So I'm going to copy and paste it. I will need it in just a moment. So going back to the homepage for NCBI, there's another feature that we're going to use this week. It's called BLAST. So it's over here on the right side of the screen. It stands for Basic Local Alignment Search Tool. What this will do is it will align a sequence of choice to a huge database of sequences that I can compare it to. So the two that we'll focus on are the nucleotide BLAST and the protein BLAST. The nucleotide BLAST, also known as BLAST-N, will compare nucleotide sequences to other known nucleotide sequences, and the protein will compare amino acid sequences to other known amino acid sequences. So I clicked on the protein BLAST, also known as BLAST-P. I'm going to paste that sequence in right here. And then I'll just go ahead and scroll down to the bottom and click BLAST. And this will begin a search, and it will compare this sequence to all other known protein sequences ever that have been entered into the database. And it will provide a list for me of the highest likelihood that alignments. So you can see that the top hits are all part of severe acute respiratory syndrome coronavirus 2. I know that is good because that is the exact same sequence that I pulled this particular amino acid from. So hold that thought. If you scroll down a little bit, you can see there's like the bat coronavirus, the bat SARS-like coronavirus, recombinant coronavirus. So there's some other viruses down here that have similar sequences. Okay, so my sequence, the one that I copied and pasted right here, this is the query. Everything that it matches to is the subject. So if I look at the query cover, 100% of my sequence is covered by the subject amino acid sequence. The percent identity is how many of the amino acids are identical. So the second one is actually what I would call a better hit, because it covers 100% of the query and it's 100% identical to the query. The e-value, again, this is the likelihood that my hit is due to a random chance. And so there is 0% chance on all of these that they align because of a random chance. This means that they're biologically significant. It means that they are absolutely related phylogenetically, that they are absolutely from the same biological source. If I scroll down to the bottom, you'll see, oh, not on this particular one. Okay, hold that thought. Okay, so if I want to compare this particular protein sequence to non-SARS-2 coronaviruses, I will do what's called an exclusion. I can exclude the SARS-2 coronavirus from my search. So I'm going to type it in. So SARS-2, and then I will click exclude, and then I'll filter it. And that will remove all of the SARS severe acute respiratory syndrome 2 viruses. And now I'm just left with some bat-like ones, which again, because this virus originally is from bats, this makes sense. And again, if I'm looking at the e-value where all these 0.0s are, this means that these are all biologically relevant sequences. If this increases, so like let's exclude all coronaviruses. So all Coronaviridae is the like general class. So if I exclude all coronaviruses, I don't get a lot of results. I actually only get, hey, it still has a coronavirus here. I'll exclude all viruses. Let's see what that does. Okay, these are classified incorrectly because they're still showing up. Okay, let's get a different protein. Okay, let's try this guy. So I'll copy and paste it. I'll go back to edit the search. I'll just get rid of this particular query and I'll replace it with my new amino acid sequence. It's a bit shorter. And then I'll search it. And you can actually exclude before searching. I want to show you what the search looks like and then what it looks like after exclusion. These searches can usually they can go up to a minute or longer, but usually they're pretty quick. Usually it's within, I would say, 5 to 10 seconds. Just sort of depends on what you are searching. Okay. So again, all my top hits are severe acute respiratory syndrome coronavirus 2. My e-value is not 0 in this case. It's still a very low number. So 4e negative 35 is 4 times 10 to the negative 35th. This is, you can think of that as like a fraction of the likelihood that this alignment is due to random chance. So something that is 1 over 4 times 10 to the 35 is a very, very, very low chance. If I scroll down to the bottom, you can see it's a little bit larger. So this one is still SARS coronavirus, and so it's probably still biologically relevant, and it is. Usually the cutoff, though, will be 1e negative 4. So like 1 over 10,000. So a 1 over 10,000 chance that my search is due to random chance. So that's the e-value. So I can actually click on this and it will show me the alignment of the query. So my amino acid sequence aligned with the subject. If they're identical, which in this case they are 100% identical, it will put the same amino acid sequence between them, or the same amino acid identity between them. One other thing is the graphical summary. So this is showing, again, it will have color-coded based on the alignment scores. You don't really need to worry about that. But this just shows the query sequence aligned to all of the subjects. And the less likely it is that it's significant, the color will change. So you can see pink is more significant than green, which is more significant than blue, etc., etc. Sometimes in the graphic summary, it will have a superfamily of proteins. So SARS-6, if I click on that, and this is a bit off topic, but I still think it's interesting. It will take you to another database where you can then look at the SARS-6 superfamily of proteins and actually learn about what they do. And so I highly encourage you to explore this website as you're doing these searches. Okay. All right, let's look at a couple other things. So if I go back, so this was the SARS-CoV-2 complete genome. Instead of clicking on this link to open up the sequence, I can actually click on NCBI virus, and that will take me to a database of all known coronaviruses in this particular case. And so these are the full sequences of a bunch of different isolates of the coronavirus. So this one's from China, all these are from the USA, There's some further on that are going to be from, there was one, Pakistan, Peru, Japan will have some, Spain has some. So this is a fun way to compare sequences or create some phylogenies of how related they are. So I'm just going to select these first, let's say like 8 or 9, and then I'm going to build a phylogenetic tree right here. I'm also going to align them so you can see what that looks like. So we'll go to the phylogenetic tree first, and it automatically creates a phylogeny of how related these viruses are to one another. And that will compare individual nucleotide identities to one another and say these two have fewer differences between them than, say, these two. If I actually want to look at those, if I go to the align feature, it will put all of them end to end together, and it will actually highlight individual nucleotide differences between them. So these are highly similar. Everywhere you see red is going to be an individual nucleotide difference. So if I zoom in a bunch, I can actually get to the letter codes, and I'm just going to go along until I find a red one. It might be a minute. I'll do this way. You can also scroll. Might be a little faster. There's different ways to visualize these things. Okay, so I'm going to try and zoom into that spot right there. Just go a little bit further. Just went out of view. There it is. Okay, so you can see that all of these have a C at this particular location, except this one has a T, and it's a thymine rather than a cytosine. And so that's just a way you can compare all of these viruses to one another. Okay, so going back to this. Okay, let's see what else we have here. Okay, so this is your Lab 11 report. I'm just going to go through it, make sure I covered everything. Okay, so I ask about E values and how you can use that to determine if your sequences align because of random chance or not. With this particular question, what I'm asking is, do you think that a longer sequence is more likely to align to something or shorter sequence? And if it's shorter, like let's say I have 5 amino acids, do you think that is going to align randomly to a lot of things or no? Okay, so I ask about the coding regions, locus tag, product, protein ID, different things. So this says pick a microorganism, so I'm just saying pick any random organism. So let's do E. coli, complete genome. And if you type in a bacteria, it's going to have a bunch of different strains. The strain doesn't matter. I don't really care. So if I click on that, it will take me to this like overview page because it's a really big genome. It's 5.5 million nucleotides. If I scroll down, I see that there's a chromosome and then there's 2 plasmids in this particular strain. I want to look at the chromosome. You can look at the plasmid if you want, but I'm going to look at the chromosome. GenBank is a database that has a lot of genomes. So it's Genome Bank is what that stands for. And then RefSeq is another database. It's reference sequence or something like that. I don't know. But use the GenBank one if they have it. So you click on it and it will take you to the same thing what I was looking at with the coronavirus is that it will have a bunch of proteins in order. So like this particular one, here's the coding sequence, here's the gene, here's the product, and then here's the amino acid sequence. The next one right here. The locus tag, again, that's just it's an arbitrary naming system for this genome only. And so I can take this particular protein sequence. If I click on the protein ID, it will take me to a page that focuses specifically on that particular protein. And it may or may not provide a link to a bunch of other databases. So this CDD is Conserved Domain Database. And so if I click on that, it will open me up to a page that will talk about this particular protein of unknown function. So I want you to think of an organism, just pick an organism and then just pick a random gene from it and try and learn as much about it as you can using the tools that we talked about. And then you're going to blast it using the protein blast. The other thing, one last thing that you can do with these, let me go back. Sorry. So one thing that you can do is you can compare two sequences. Rather than comparing it to all known sequences, I can compare two sequences of my choosing. So let me just take this random protein and I'll go to the protein blast. I click align two or more sequences and I can paste the first one in there, and then I'll go to this one and I take this sequence, and it doesn't matter if it has numbers in it, it will exclude them automatically. So up here's the coronavirus gene, and then up here is the E. coli gene. So now if I blast them, it will compare the sequences to one another to see if there's any sequence similarity. In this particular case, no sequence similarity was found, meaning that they are not related at all. And that's expected because one's a bacteria and one is a virus. And so I want you to explore that a little bit. You can also do the same thing with nucleotides. I don't think I included it in the lab report, but we will definitely be doing it next week. And the last question is identify at least one feature from the database that I didn't talk about in this video. Just click on stuff. Just see where it takes you, see what you get. I don't know what a lot of these things are, and so I highly recommend just clicking on random links until you get to something fun and see what you can learn. Okay, so that is that. Again, I am available during the days, and so if you email me with your questions, I'm also happy to do video chats. Again, I want to be as accessible as possible, and I still want you to be able to learn things. However, I don't want you to overly stress about this. So yeah, please let me know if you have questions, and stay safe and healthy.